Hello, this is Dr. Panigrai. Welcome to my another session. In this session, I have a talk about the overview of transcription in prokaryotes. This is the introduction to transcription in prokaryotes. So let's start. What is transcription? So the idea is very simple. Transcription means the synthesis of RNA from DNA. This process is known as the transcription. And such RNA which is translated into protein, whatever sequence, nucleotide sequence present in the RNA, such nucleotide sequence is copying or translate into in a form of the sequence of amino acid, and such process is known as the translation. The total transcription and the translation process collectively known as central dogma of molecular biology. And the gene or the segment of DNA which expressed itself in a form of protein. That's why the transcription and the translation, these are the two main events of the gene expression. So gene expressed by the two process, one is the transcription, another is the translation. Suppose that this is the strand of DNA. Suppose that this is the strand of DNA. So these DNA strand which synthesize the RNA strand. So it needs some protein, and such protein act as the enzyme. That's why this enzyme which synthesize the RNA strand, and such enzyme is known as the RNA polymerase. So the transcription process which need the RNA polymerase enzyme. By the binding of RNA polymerase enzyme, the DNA strand which unwind, and it forms the transcription bubble like structure. So this is known as the transcription bubble. And out of these two strands, one strand which participate in the transcription process, it means one strand actually act as the template strand. Suppose that this is the RNA strand. So this RNA molecule which is synthesized from the blue DNA strand, such strand is known as the template strand. Template strand. The RNA strand, the whatever sequence, whatever nucleotide sequence present in RNA which is complementary to the template strand. But the nucleotide sequence of RNA which is similar with the another strand, and such strand, the red strand, is known as the sense strand. Such sense strand also known as coding strand. This is known as coding strand, or also known as the positive or plus. This is plus strand. And complementary strand of sense strand which is known as antisense. This is known as antisense strand, or also known as non-coding strand. So this is very important. So the template strand which known as the non-coding strand. Such non-coding strand actually participate in the synthesis of RNA. So RNA synthesis takes place in the direction of 5' to 3'. And this is the DNA strand having this is the 5' to 3', and this is the 3' to 5'. So this is the 3' to 5', and the RNA synthesis which takes place from the 5' to 3'. So the synthesis which takes place, or the synthesis of RNA which need the enzyme which is known as the RNA polymerase enzyme. Such RNA polymerase enzyme binds on the site of the DNA or specific site of the DNA and unwind the DNA strand, and which forms the transcription bubble. Such transcription bubble which move or which read the template strand in the direction of 3' to 5'. So RNA polymerase molecule which read the template strand 3' to 5' direction. And the synthesis of RNA which takes place from the 5' to 3' direction. This is the synthesis of RNA. So the total process of transcription, it needs one enzyme which is known as the RNA polymerase, and such RNA polymerase, this is the one protein which actually participate in the process of transcription. That's why such RNA polymerase is known as the transcription factor. In case of prokaryotic cell, this is the single transcription factor which involved in the process of the transcription. And we need some of the ingredients like nucleotide triphosphate for the ribonucleotide triphosphate for the synthesis of the RNA strand. So now we have the template strand or the antisense strand, and we have the sense strand or the coding strand. So the RNA polymerase which recognize the specific sequence which present on the DNA starting point of the transcription unit, and such sequence is known as the promoter region. So the DNA, suppose that this is the DNA and the RNA polymerase, suppose that this is the RNA polymerase enzyme which binds to the particular specific site of the DNA for the process of transcription, and this site is known as the promoter site. Promoter site, it has some special or the specific sequence which actually recognized by the RNA polymerase, and such sequence actually present at the region of -10 and at the region of the -35. So now what is -10 and the -35? The side from where the transcription start, and such point actually known as the, suppose that this is the start point of the transcription, and such point is known as the +1. So this is the starting point, this is the start point, this is the start point. So This is the DNA strand and it has the start point. This is the start point. Such start point which is rich in A or G, adenine or the guanine, and which is indicated as +1. After start point, the region is known as the downstream. And prior to the start point, the region of DNA which is known as the upstream element. So the RNA polymerase which come and sit on this portion, or the RNA polymerase which recognize the -10 and the -35. So the nucleotide sequences which present from the start point or after the start point towards the downside indicated as +2, +3, likewise +10, likewise +20, like this. And the sequences or the nucleotide base pair which present just left to the start point, which is indicated as -1, -2, like this, -10 and -35, like this, so on. So the distance between the -35 to -10, which is the 7 to 19 base pair, and the distance between from the -10 to start point, which is 6 to 7 base pair. There is no, you have to remember, there is no 0. The starting point is +1. There is no 0 is assigned on the DNA. There is no nucleotide is assigned on the DNA. So we have two important sequence at the region of the -10 and at the region of the -35. Such -10 region and the -35 region which has the 6 base pair sequences, 6 base pair. So -10 which has the Ta8, thiamine, adenine, thiamine, adenine, adenine and thiamine. And at the region of the -35 there is the Ta, Ga, Ka. So thiamine, thiamine, guanine, adenine, cytosine and adenine. So these are the 6 base pair sequences which is present at the region of the -10 and the -35 region. These sequences also known as the consensus sequence, or also known as conserved sequence, because it conserved generation to generation. The sequence of -10 region, which is also known as the Pribnow sequence. It is named as the one of the discoverer, and such Tataite sequence also known as the Tata box. This is very important because the melting actually start at the region of the -10 region. So the promoter, which actually composed of number of sequences of the DNA, and such sequences of DNA actually recognized by the promoter, the RNA polymerase enzyme, and such RNA polymerase enzyme actually recognize two particular sequences or the specific sequences which actually present at the region of the promoter site, and these are known as the Pribnow sequence, and another is the Tagaka. So many genes are there, and very rare of gene having such type of sequences at the specific site. But most of the promoter, they deviate from such typical type of promoter. So some have the -10 sequence and the Tata box, and some have the -35 sequence. But the RNA polymerase also recognize some extra sequences. Those are present upstream of the -35, and such sequences are present far away from the -35 sequence. Suppose that these sequences are present here, and these are known as the upstream element. In some of the gene which have, this is the start region, which have only -10 or a Tata box sequence, but there is no -35 sequence. In this condition, the RNA polymerase recognize some of the sequences which are present just prior to the -10 towards the upstream region, and such sequences are known as the extension. These are known as the extension element. And some of the gene, this is the start point, they have the Tata box region at the -10 region, they have the certain sequences at the -35 region. But the RNA polymerase, which bind or recognize some of the sequences, those are present downstream of the -10 region, downstream of the -10 region. Suppose that these sequences are present in between the -10 to 1 of the start point, and this is known as the discriminator. This is known as the discriminator element. So each promoter should have the two important consensus sequence, Tata box at the region of the -10 or stag at the region of -35. This is the typical structure of promoter. But such typical structure may present very rare in some of the genes, but most of the genes, they differ from such type of typical promoter. And there may be the upstream element, there may be the extension element, or extended, also known as extended element, or discriminator. So these sequences are the promoter sequences which actually recognized by the enzyme is known as the RNA polymerase. So in case of prokaryotic cell, such RNA polymerase enzyme is very important because it has two roles. The first role, to addition of 1 and 1 nucleotide at the direction of 5' to 3' for the synthesis of the RNA, and in other side, such enzyme also play the work of proofreading or editing process. It check out whether the right nucleotide added or not. If there is mismatched nucleotide present, wrong nucleotide add to the newly synthesized RNA, then cut out and remove from this site. So proofreading process, proofreading works, as well as the synthesis work or the addition of nucleotide, new nucleotide to the RNA, which actually takes place by the RNA polymerase enzyme. So in case of prokaryotic cell, the RNA polymerase enzyme, which is composed of beta and beta prime subunit, and these subunit forms the active site or active clip of the protein of the enzyme. And there is another subunit, those are the alpha. So two alpha subunit are present. These two alpha subunit, they actually assemble the different subunits. So the alpha subunit actually role to assemble the different subunit of the RNA polymerase. And the presence of another subunit, which is omega, and the presence of omega subunit, this is the controversial. So this is the controversial. So now you can say there are two alpha subunit and one beta, another beta prime subunit. This subunit, it forms the core enzyme, which forms the core enzyme. And such core enzyme in association with another factor known as sigma factors, it works. So the core enzyme in association with sigma factor, it forms holoenzyme. So this holoenzyme or such sigma factor actually recognize the -10 Tata box region or the -35 sequences, and which actually help for the binding at that site. So I have another more video regarding the sigma factor, and which actually control the regulate the binding of the RNA polymerase enzyme. So these holoenzyme which recognize and bind to the promoter site. So the core enzyme or core part of the enzyme in association with sigma factor, it works. So in prokaryotic cells, so many sigma factors are there, and for a particular promoter, a particular type of sigma factor is present. The sigma factor 70 is general for the E. coli bacteria. So sigma factor 70, this is the general sigma factor, and such 70 in association with the core enzyme, it forms the holoenzyme, and which find out the promoter region and the specific sequence on which it will bind and melt the strand and move or read the template strand and continuing to the synthesis of RNA strand. This is the process, and the synthesis process is continuing from the start region and up to it reaches to the terminator sequence. If the terminator sequence is approaching, or the presence of terminator sequence which sees the addition of new nucleotide to the 3' end of the RNA, and this is known as the termination. So the total process of transcription you have to studied under, one is the initiation, another may be the elongation, and the termination. So the total mechanism we have to study under initiation, elongation, and the termination. So we have three different video regarding the initiation, elongation and the termination. And also we have the regulation of the transcription in prokaryotes, and we have another session regarding the sigma factor, regarding the RNA polymerase, regarding the Tata box. So many series of videos are present in my channel regarding the transcription in prokaryotes. So the core enzyme in association with alternate sigma factor, it works. So same core enzyme which bind to a particular sigma factor and which recognize the specific promoter of the specific gene. So same core enzyme again bind with another sigma factor and which again searching or find out the another promoter of the another gene. So after the initiation process, such sigma factor dissociate from the core enzyme. So many mechanisms, so many concepts, so many models actually proposed for the initiation, for the elongation, for the termination. So I suggest you please click on my another video or subsequent video to get the whole portion of the transcription in prokaryotes. Thank you.